So our speakers today are from the Research Institute for Fragrance Materials. Insiders call it RIFM. And RIFM assesses the safety of fragrance ingredients in accordance with the most current internationally accepted guidelines. And since 2014, it's using an updated set of criteria designed to incorporate the most current science while avoiding animal testing. And our speakers today are, first we will hear from Dr. Nikaeta Sadekar. She leads the Respiratory Endpoint for Safety Assessment and Research efforts at RIFM, and she collaborates across the scientific community to develop RIFM's respiratory science. Then we'll hear from Dr. Miwa Na. She leads the Skin Sensitization Endpoint at RIFM, and she conducts risk assessment and research projects to address the potential of a fragrance material to induce skin sensitization. So now we'll go over to Nikaeta. And again, if you have questions, please type them right into the chat and we will answer those after both speakers. Thank you. Over to you, Nikaeta.
Thank you. I'll start with sharing my screen.
Perfect.
All right, I think Riddhi will have to share it for me. Sorry.
Okay, no problem. Riddhi, Riddhi is doing all the magic behind the scenes. So we'll be great. We'll be in presentation mode. And here we go. Thank you, Riddhi.
Thank you so much. All right. Thank you, Laurie. That was a very kind introduction. Hello, everyone. So for today's talk, again, fragrance is a very interesting topic. And most of us use scented products practically every waking moment of our lives. You may think surely the consistent exposure to fragrances must add up and wonder if it is safe. I'm a toxicologist, and the most basic lesson you learn as one is that the dose makes the poison, which means that the exposure is an important part of a safety assessment. At the Research Institute for Fragrance Materials, we work towards ensuring that the overall use of fragrances is safe. Next slide, please. For more than 50 years, RIFM has worked to fulfill our core purpose, which is to build universal acceptance and trust in the safe use of fragrance materials through applied science and research. We are a nonprofit organization. If you can click through. We generate data, analyze, evaluate, and distribute scientific data so that the consumers can enjoy scented products worry-free. Next slide, please. We have the expert panel for fragrance safety. It is an international group of academics who provide the scientific authority and objectivity to RIFM's work. This panel has been in existence for over 50 years now. And for more information, please visit the website shown below. Next slide, please. All the panel-approved RIFM safety assessments and research is published in peer-reviewed scientific journals. Full text access to RIFM's work published in the Food and Chemical Toxicology Journal is available at the Fragrance Safety Resource Center. Click through, please. You may visit this resource center at fragrancematerialsafetyresource.elsevier.com. And you may also subscribe to receive regular updates. Next slide, please. Fragrances have unique properties, and that sometimes warrant special considerations for safety evaluation. These compounds are volatile and organic in nature. They are presented with odorant properties, and as such are used in much smaller quantities in the final products. Their volatility significantly alters their exposures, but RIFM balances the use of conservative and practical methods for risk assessment to ensure their safety. The underlying assumption is that 100% of the fragrance is available for exposure from a product. Next slide, please. So today I will talk briefly over the RIFM safety assessment process. And as the respiratory endpoint lead, my talk will focus more on the local respiratory toxicity endpoint assessment at RIFM, which includes the safety assessment for fragrance ingredients and the natural complex substances. At the end, I will also briefly introduce the respiratory research program. Next slide, please. So as I was saying, the exposure is an important part of any safety assessment process. The human exposures to fragrance ingredients from a variety of product categories are calculated by the RIFM aggregate exposure model. Click through once, please. This model generates values for inhalation, oral, and dermal routes of exposure. The categories that are included to calculate these exposures include the cosmetic, personal, air, and household care products. Click through once, please. It is a probabilistic model based on real-world data, and the RIFM safety assessments use the 95th percentile, the high-end user exposure value. Next slide, please. So using these exposure values, we cover in total 7 different endpoints, which include genotoxicity, repeated dose toxicity, developmental and reproductive toxicity, skin sensitization, photoirritation, photoallergy, local respiratory, and environmental effects. Next slide, please. So all the endpoints generally follow this 4-step process, but depending on which endpoint, the process can be slightly different. These 4 steps form the fragrance safety evaluation criteria that was published in 2015. It prioritizes using existing data over testing and generating new data, which is only considered as a last option. With a focus on the respiratory endpoint, the objective is, next slide, please. The objective is to analyze for the point of contact toxicity via inhalation exposure. In other words, we evaluate for effects in the respiratory tract. Next slide, please. So as for the 4-step process, the first step involves using data available on the target material. If it is sufficient, then the safety assessment is completed. If it is insufficient or limited or no data are available, then we move on to step 2. Here, we search for a chemically similar material that can be used as a read across for the target. If sufficient data is not available, then we move on to the next step. However, if sufficient data is available, then we complete the safety assessment here. Sorry, next slide, please. Now, before I move on to the process, the next step in the process, I would like to highlight how we determine whether a study is sufficient. It is based on several key considerations. First, we do a thorough literature search for inhalation exposure studies that contain data for local respiratory effects and ask the following questions. Does the study follow any of the guidelines? What is the duration of exposure? Which animal species or strain is used? What is the mode of exposure? What is the range of exposure concentration? What effect level is identified? What is the basis for identification of those values? Are there any observations pertaining to local respiratory effects? Answering these questions can help you make an expert judgment whether the data is sufficient to complete the assessment. And in most cases, the available data are insufficient. The slide that we are seeing right now, this is what we do. If we do find a study with enough details to make an evaluation, click through once, please. We identify the no observed adverse effect concentration. Click through once. Then we normalize that NOAC value to species-specific minute ventilation value and lung weight. Click through once, please. And at the same time, we use the human inhalation exposure value for the fragrance ingredient under consideration and normalize it to the human lung weight. Then we calculate the margin of exposure by taking the ratio of the two normalized values. And if it is over 100, then we complete the safety assessment. Next slide, please. Also for the steps, when we come to step 2, selecting an appropriate read-across analog involves an endpoint-specific consideration. Now, I've adopted this slide from one of my colleagues as it wonderfully lays down the 3-pronged approach for validating our read-across choices. So click through once, please. Sorry, no, go back. I thought I had animations on this slide, pardon me. So I have in this slide, you see the first box with the structural comparison. We look for the similarity in the structures and also consider the functional groups and the reactivity of the chemical. As a second step or second consideration, we look for the differences between the physical properties, which would involve the boiling point, melting point, vapor pressures, log KOW values or solubility, water and lipophilicity, and also skin absorption in some cases. And lastly, we also use the available in silico tools such as the EpiSuite and QSAR model to find additional insights for optimal comparison. And again, we confirm if the available data on this read across is sufficient for the assessment. Next slide, please. Now for the respiratory endpoint, we often find ourselves at the step 3. Using an exposure-based waiving approach, we find as an effective animal alternative technique as about 75% of the materials in our inventory are cleared at this step. This involves applying the concept of inhalation threshold of toxicological concern, or inhalation TTC, as proposed by Philip Carthew in 2009. In a moment, I'll talk more about this concept. But in extremely rare cases, if the endpoint assessment cannot be completed using step 3A, then we follow step 3B, where we use an in silico model, the multiple path particle dosimetry model, or the MPPD model, to refine the inhalation exposure value. Next slide, please. So for the concept for using threshold of toxicological concern, it means that the level below which the human exposure to a substance is considered to be of negligible risk in the absence of chemical-specific toxicity data. And as part of the 4-step process, we use TTC as an exposure-based waiving approach in the absence of data for a fragrance ingredient. We note that the inhalation exposure for over 90% of the materials in our inventory are below the most conservative Kramer Class 3 inhalation TTC limit of 470 micrograms per person per day. This was established by Carthew et al. In 2009. Next slide, please. So as you can see, the stepwise approach has proven to be of great importance for utilizing animal-free methods for safety evaluation of fragrances. And the most sustainable aspect is that all the endpoints have benefited from this approach by saving over 500,000 animals. As such, the read across and the TTC form a strategic part of the RIFM safety assessment program. Next slide, please. Now the final step in the process, step 4. In even extremely rare cases, if the respiratory endpoint assessment cannot be completed using the first 3 steps as highlighted before, then the material will be sent for testing, which is this step, the 4th. So far for the respiratory endpoint, we haven't had a scenario which required testing in order to clear a material. Next slide, please. And so as you've seen for the discrete fragrance ingredient safety evaluation, we follow a very similar process to evaluate the natural complex substance mixtures. Again, every endpoint has a unique approach embedded in the process. For the respiratory endpoint, the 4 steps are divided under 2 categories: whole oil evaluation and component evaluation. The slide that you're looking at now outlines the whole oil evaluation, which involves the first step, looking at data available on the whole oil. If suitable data is not available, then we use the exposure-based waiving approach as step 2. Here, the chronic aggregate inhalation exposure for the whole oil is compared to the Kramer Class 3 TTC limit of 470 micrograms per person per day. Next slide, please. When the whole oil exposure is above this threshold, Then the evaluation is based on individual components in the NCS. These steps mimic the criteria I just introduced you to for the discrete ingredient safety assessment, where again, the available data for target and the analog is prioritized over exposure-based waiving approach. The final step in the process here uses refinement by MPPD model when the derived inhalation exposure to any component is above their respective primer class TTC limits. Next slide, please. Although our efforts are to maximize the use of existing in vivo data, the biggest challenge for the respiratory endpoint is the limited availability of existing data and the lack of standard validated models for testing. With limited inhalation studies available, the possible read-across analog options are also limited. And it is for this reason that RIFM has been focusing on research for the respiratory area, specifically for developing new alternative methodologies. Next slide, please. Currently, we are involved in identifying and developing new approach methodologies. Click through once, please. So these involve looking at local respiratory effects and assessing respiratory sensitization. Click through once, please. We are also invested in enhancing the inhalation TTC by expanding the Carthes dataset. And for the last one here, the unique odorant property of fragrance ingredients means very little material is needed to impart an odorant aspect to the products. And to that effect, we are working on a project to detect odor thresholds in humans. Next slide, please. All these projects are carefully designed with the help of many of our collaborators. We believe that collaboration and teamwork are the current climate for respiratory research. Click through once, please. And all of this is possible only because of the support from an amazing team at RIFM. With that, I thank you all for your time, and I'm happy to take any questions you may have.
Great. Nikaeta, thank you so much. Now we'll